Good morning. We want to welcome you to the Center for Iron and Heme Disorders Interviews with Giants in Iron series. We're so privileged to have Nancy Andrews speak with us today. Um, I've known Nancy for a lot of years, and Nancy has been in the iron field for many years. I want to just briefly introduce her, and then we will turn the time over to Cade Loveridge, who is an MD PhD student here at the University of Utah, who will conduct the interview. So Nancy received her bachelor's degree and master's degree in molecular biophysics and biochemistry from Yale. And then she went on to MIT for her PhD. From there, she went to Harvard for her MD, and she did an internship and residency in pediatrics at Boston Children's Hospital and a fellowship in pediatric hematology and oncology at Boston Children's Hospital and Dana-Farber Cancer Institute. There are so many accolades I could mention for Nancy that I'm going to just skip them and do the interview, because that, I think, is the sweet part of this opportunity to talk to her. And I will turn the time over to Cade. And thank you guys. And Nancy, thanks so much. I know that it's a busy schedule, and you guys have no like IV fluid in Boston, according to the
True
news.
enough. True enough. Well, thank you, Diane, and thank you in advance, Kate.
Yeah, I'm very excited for this interview, and I just wanted to start with just kind of your background and family, if you wouldn't mind just kind of giving us that perspective.
Certainly. So I am, other than 14 years I spent in North Carolina, I'm a lifelong Northeasterner. I grew up in Syracuse, New York, and my father was a lawyer there, one of a long line of lawyers, and my mother was a social worker. My father's not living anymore, but my mother is. And I have three younger brothers who, none of whom are in science or medicine. No one in recent generations of my family, my age or older, has been in science or medicine. But both of my children are, my son is an MD PhD student at Harvard and MIT, and my daughter is a thoracic surgery fellow here in Boston at Massachusetts General Hospital. And my nephew is in training in pediatric neurology. And so I think from here on, there will be more science and medicine. But I went to an inner city high school in Syracuse, and I think had opportunities that were less academic and more learning about the world in that place. And I think that's served me well for many years. And beyond that, my academic history, I went to Yale for college, and then I was an MD PhD student at Harvard, receiving my PhD from MIT and my medical degree from Harvard, and was in Boston for most of the time after that, but spent 14 years in North Carolina, 10 of which I served as Dean of the School of Medicine at Duke University. And I guess I should also say that I've closed my lab now more than 8 years ago, which is kind of hard to imagine. But it was largely because of my administrative roles. I just started to think that it was going to be difficult to give my students and postdocs the attention they deserved. And I thought I might have a move coming up. And I ultimately did. I left Duke to come back to Boston, and I didn't want to put people through that. And I realized that there are a lot of young people who needed the funding for research more than I did.
Okay.
Well, thanks for giving us that kind of overview. And that's a perfect segue into, I mean, you had a really amazing scientific career. I would love if you could kind of tell us a little about your scientific career and also kind of how that led into iron metabolism. And I'll
Certainly.
just, however you want to do
So
that.
it was, I suppose some might say, a peripatetic scientific career. That was not on purpose. I did my PhD with David Baltimore at MIT, and I joined his lab because I was interested in what they were doing in immunology. The first project I had in his lab towards my PhD thesis ended up being stopped because it was based on an observation that was misinterpreted. And when it became clear that the reason for pursuing that line of inquiry wasn't correct, I switched and I worked on poliovirus and did my PhD work in David's lab, following up on what he had done as a PhD student 20 years before. I didn't stay with poliovirus when I went to, actually, I skipped a very important part, and I'll come back to that last. But when I finished my clinical training and became a fellow in hematology oncology, I worked with Stuart Orkin initially on red blood cell transcription. I'll get in a moment to how I got into iron, but first I want to say that my initial real scientific experience was as an undergraduate at Yale with Joan Stites, and that was a formative experience, and perhaps probably the most formative of my career. So I shouldn't have skipped it. I worked on transcription of a phage and figuring out where the transcriptional start site was in a bacteriophage in Joan's lab. And then I worked a little bit on the small ribonucleoprotein particles that she became famous for. I was in Joan's lab at the time she made the brilliant discovery that these small particles were part of what we now know as the spliceosome. And it was a very, very exciting time to be there. And it was important because Joan is an amazing scientist. I learned a lot from her. And I also learned that MD PhD programs existed because her lab was a magnet for MD PhD students. And that changed my life. I was going to be a chemistry or biochemistry PhD student after college. And when I realized I could do both medicine and science and find ways to contribute at the interface, that really changed things. So that was the early part. As I said, I was a fellow. I did my postdoc fellowship research in Stu Orkin's lab, working on red blood cell transcription. And when I left Stu's lab, I realized that it would be very hard to compete in that field. I got my first NIH grant working on red blood cell transcription, but the questions were being addressed by many labs around the world. And I was looking for something that would be a little bit more open, where I could ask bigger questions, where I wouldn't feel like there were many people doing the same thing. And I was very fortunate that when he was a medical student and I was a postdoc still, I met Mark Fleming, who was a Harvard medical student who, and I think he talks about this publicly, had a relative with hemochromatosis and was very interested in iron. And Mark convinced me to choose iron as my field. So not long after I opened my own lab, within a couple of years, with Mark, really brilliant guy, we started working on iron. And Mark in particular wanted to use classical mouse mutants, spontaneous mouse mutants that had mutations that we knew affected their iron physiology, but that weren't in genes that had yet been identified or had not yet been shown to be in particular genes. He knew that that was a very rich way to learn about iron metabolism. And so together we learned mouse genetics and went about finding genes that were mutated in mice with iron disorders. And that was really how I got my start.
Yeah, I just a comment from the peanut gallery. I think you guys should have all the credit for starting the utilization of mouse mutant genetics to segue into iron metabolism in the field. You guys were the pioneers in it, I think. It was really profound and affected the direction of iron research for a long time.
Well, thank you. And I have to credit Elizabeth Russell in particular, who decades before at the Jackson Laboratory realized that those mice were interesting, realized that they probably had iron problems, and just made it possible by purifying colonies and keeping them and studying them. And I sometimes think that she and others in the 1960s and 70s, they did the important work, and we just put names on the proteins or activities that they knew had to be there.
Yeah, a good mouse geneticist goes a long ways.
Yes.
Good.
Well, so you kind of answered the first part of my next question, which was, and you can expound on this more too, but what iron research was like when you began your career. But, as you, ran your lab and made all these really profound discoveries, how did you see the field change? Yeah.
So when I first started, and Diane should weigh in on this if she doesn't agree, because I think she may have a better perspective. But to me, coming from the outside, coming from a red blood cell transcription background, it looked to me like a lot of the attention in the iron field was on iron regulatory proteins binding to what had been identified as RNA iron regulatory elements, really important work, or work in yeast, as Jerry Kaplan's lab was doing, or in bacteria. But it was still unclear, or work on transferrin structure and ferritin structure and so on. But what seemed to be wide open and not completely understood was how iron got into eukaryotic cells and how iron got out of eukaryotic cells. And so reasoning that if we ultimately wanted to understand hemochromatosis or contribute to the understanding of hemochromatosis, we'd have to first know how iron gets in and out, because it's a problem of too much iron in the body. And so that's where we focused in the beginning, was actually how iron gets into the body. And at the time, people had ideas that turned out not to be correct for mammals. I'm talking about was much better understood in yeast and in bacteria. But in mammals, it was kind of a complete black box. And so that was where we started out.
Yeah, I 100% agree with that. Yeah.
So I know this is a tough question, but looking back, is there one or two discoveries that you made that maybe are your favorite scientific contributions?
I think really two. One was when we realized that we had probably found the first mammalian non-heme, actually, to a first approximation, the only, although other proteins can do it, non-heme iron transporter that brings iron into cells, that at the time was called NRAMP2. It was a known gene, but of uncertain function, and had not been implicated as an iron transporter. Later called by some competitors of ours, DCT1 for divalent cation transporter 1. The name that we and others in the iron community gave it, DMT1 for divalent metal transporter 1, stuck, and then the genetic name is SLC11A2. But when we realized that we had found a mutation in the NRAMP2 protein that was convincingly evidence that NRAMP2 was the iron transporter, I think that was the most exciting moment. We were looking for the gene by mapping in mice at a time when the mouse genetic map was still really crude. And we hit it lucky that a colleague nearby knew that a gene called NRAMP2 was in the right region of the mouse chromosome. In those days, that was really lucky, because it could have been very far away and still looked like it was in the right region. And we realized that that was a strong candidate because it had an ortholog in bacteria that was involved in manganese transport. And so I think it was in bacteria. And so that was, maybe it was yeast, but that at any rate, manganese is enough like iron that was a great clue. And I remember when the technician in the lab who was working with Mark Fleming called me and read me the amino acid sequence, and I was following the normal amino acid sequence on a piece of paper. And I realized he was reading from a sequencing gel, just translating the codons. And I realized that there was a really significant change in the sequence. And so that was very exciting. And I think that the other time was when we realized that, or at least we thought, and we turned out to be largely right, that altered expression of hepcidin was really important in the anemia of chronic disease, a very common medical condition. And we made that hypothesis pretty early, and it took a while to accumulate enough evidence to convince everybody, evidence from our group and from others. But I think now that's pretty well accepted that inflammation leads to increased hepcidin expression, leading to much of the picture that you see in the anemia of chronic disease. So I think those were the two most fun parts, but there were a lot of fun parts over the years.
Yeah, and I study a DMT1 homolog Plasmodium and just I love that protein. So it's fun kind of hearing some of the foundational discoveries. But so I want to ask another kind of science question and then circle back to some of the influential people, both mentors and trainees. But I guess I want to ask you, what is, in your mind, the biggest unanswered question in the field of iron metabolism?
In a broad sense, it's what's going on in neurodegenerative diseases. And in different neurodegenerative diseases, there are hints that iron homeostasis is altered either as a cause or as a consequence. And I don't think we understand that very well yet. And I think it's going to turn out to be really important. But it's a wide open area. We had in my lab ideas about what might be going on, but we didn't take it all the way to fully prove it. But the role of iron in the nervous system in general, but most particularly what's going on with iron in neurodegenerative diseases, to me, is the most interesting.
Very, very interesting
brain
stuff.
frontier. Yeah, has been a question a long time.
It has.
And a difficult question to address, actually. I can remember in the early, I think 80s, maybe 90s, where we would keep talking about the blood-brain barrier and how you would try and get iron into that area to sufficiently fill the needs of the cells in there. And it was a hard question, and still is a complicated question.
I think that's right. I think that's right. I just had a thought, something that I was going to mention. About it being a hard problem. Coming back in the car today with my son, driving back with him from a trip. He's a scientist, as I mentioned, or becoming a scientist. I think he is already. I said that the best advice that I remember ever getting early in my career was to choose a hard problem and to stick with it and to do whatever it took to answer it, but to stick with it, because if it was an important problem, then I could be sure that finding the answer would really matter. And that's what we did. That was my philosophy starting out in looking for DMT1. And it took a long time because mouse genetics wasn't very far along at that point. Actually, it took about 2 years, which maybe wasn't a long time, but it felt like it. And I was just starting out. And I think a lot of people around me thought that I might be sacrificing my career if it didn't work out, because I was putting so much attention into that. But I think that's where having a colleague like Mark Fleming, who was as enthusiastic as I was, maybe even more so, made a huge difference, because I think we kept each other going and we got there.
I think that's true for a lot of the community of investigators in iron. They had a collaborator and the two of them together really drove science. Yeah. And then that community together, as we were a small community early on, we helped drive each other and really had good conversations that shared observations, and we weren't afraid to talk about them.
And that was wonderful, coming from a field that was very competitive and kind of overpopulated. It was wonderful to come into the iron field and have great colleagues who would talk about things and who um, just created a culture that was much better than where I had come from,
Yeah.
in
Yeah.
my view.
My view too.
Well, so you already mentioned a lot of influential mentors. If you wanted to mention any more, or as well as colleagues, as well as any influential trainees. Yeah.
I think I've probably mentioned most of them. Mark is probably my most influential trainee. He's now been for, I don't know, probably 15 years, something like that, the chair of the Department of Pathology at Boston Children's Hospital. And so has been influential in that way. He's continued to have a lab and continued to make contributions to understanding iron, particularly related to red cells, but also put a lot of energy into building a very strong department and into collaborations with many people outside of the field of iron who benefited from having a smart pathologist on their side.
Um, are there any stories that come to mind of just either happy or funny interactions with the iron community?
Well, the first thing that comes to mind is I remember receiving to review for Proceedings of the National Academy of Sciences, paper written by Sophie Vallon's lab in Paris, where they had done a knockout of a gene that they were interested in and got a very severe iron phenotype, hemochromatosis phenotype in mice, and realized as they worked this up that they had accidentally knocked out the hepcidin gene. And that was, while there had been hints that hepcidin was related to iron before, that was kind of the hit it out of the park experiment. And they didn't mean to do it, but they knocked out hepcidin accidentally. And I remember getting that paper to review and reading it, and others might have been skeptical, but I was right away sure that they were right. I thought they had made a terrific case, and I didn't know anything about hepcidin yet. It was just being discovered simultaneously in a few labs, but Sophie's paper, I just read it and it suddenly clicked, and I thought this is going to be hugely important. So I wrote in my review, I could ask for more experiments, but I think you should publish this as soon as possible, so I won't. And I wish that I got reviews like that more often, but I really felt strongly it needed to get out in the scientific literature, and we wanted to work on it, and we couldn't work on it until it was published. And so I thought that it was really important, and that hopefully it made Sophie smile when she got that review. I suspect it did. I'm not sure the other reviewer or reviewers were quite as simple in their remarks.
Yeah, it's always good when you recognize something, um, such a big deal, just giving somebody their flowers for making that discovery.
Yes.
And so I guess I feel really grateful that I get to help out with these interviews and interview, like Diane has called it, giants in iron metabolism. And I have benefited so much from the community that you and others helped make. And so I want to ask, somewhat selfishly, and for other young scientists listening, what advice would you give to researchers in the iron metabolism field? As we try to carve out our niche and also make impactful discoveries.
I would say iron for me, and I think this is still true, I hope it's still true, always had a lot of uncharted territory. It meant that when postdocs left my lab, I could let them take with them whatever they were working on because there was always something else to go to. Obviously, a lot has been accomplished, and there's less uncharted territory than there was, but there still is, I think, a lot. And so think about the questions, try to think about the questions, and this isn't easy, that um, others haven't asked yet, the things that are interesting that need to be explained that others haven't asked yet. Or as an alternative, choose a problem that is still actively being worked on. I think ferroptosis is fascinating, and I think that there will undoubtedly be more to be discovered there, although that's getting a lot of attention. And iron and neurodegenerative diseases. But think about the things at the fringes. Those to me were always the most interesting. Less competitive, maybe of less interest to others to start out. But again, if there's an important question in there, then it's worth doing for sure. And what I was doing in iron seemed to a lot of people to be pretty fringe when I started out, but we got there.
Which is, yeah, just wild for me to think about people considering that fringe, given that it's so foundational to our understanding of iron metabolism. So, yeah, thank you so much for answering the questions. Those are everything that I have, super informative. Great to hear about more detail about your career and research, and I really appreciate it.
Well, thank you. I appreciate your doing this, and you've been a great interviewer.
Yeah, just a couple other things that I wanted to chime in on and ask about, just in the relevance of maybe comments for the community that might watch this. Do you have advice on how to balance your career and family?
So my philosophy there has always been family first. And that helps. If I have to choose, I choose family first. Thankfully, I've rarely had to choose, and that I've been very lucky in that. My husband and my kids have been healthy and easy overall, not always, but overall. And my parents haven't needed a lot of care from me overall. And I think I've been very lucky that while I know what I would choose, there's probably only been like one time in my career when I really had to make that choice. It was the one that comes to mind, at least, was I was I'd finished my term as dean at Duke, and Duke started a lectureship in my name, and I was supposed to present someone who I had known for many years, an outstanding scientist, as the first inaugural lecturer for that lecture. And I think it might have been the day before or that morning, 4 a.m., I heard that my daughter was in the hospital in Boston. I was in North Carolina, and with a pretty serious condition that was going to need immediate surgery. And so I flew up to Boston, knew that I was going to get stuck in a blizzard there because the weather forecasts were terrible, and knew that I would miss being there for that lecture. But it was, same decision I would make anytime.
Yeah, absolutely. And I always tell young people coming up in it, you know, you have to make that priority, but you're never, for me, you're never sorry when you choose the family piece first, because that's the one you reflect back on often.
Yes. And at the end of my life, that's what will really have mattered. But I was very lucky that with wonderful people around me, both at home and at work, my husband particularly at home, and a lot of great colleagues at work, it worked out. Everybody's got their own way of doing it, and it helps to be lucky and not have difficult family problems to have to deal with. But it can work.
I agree 100% with that. I'm trying to think of other fun stories that might be in the iron field that you have hidden in your brain that might be of interest to the community. I mean, there's a lot of funny stories that are around about, but I was trying to reflect on early iron meetings where, although we talked about being highly collaborative, sometimes they were kind of contentious. My favorite stuff that I remember were things, maybe I'm going to give credit to Prim Ponca, who had no fear in challenging what someone was trying to present as factual. And
That's true.
Yeah, he had, and I think that drove better science, and that the communication between people, we knew that there wasn't a personal attack, but it was trying to get at the right answers. And
Um,
yeah,
I, I think that's right. He sometimes was critical of young people, and I had a bit of trouble with that. I think, I felt it was fine to be attacked for me, but sometimes but you reminded me of one thing though. I don't know if it's funny, but I guess you'd have to, if you have a perverse sense of humor, maybe it's funny. But I remember the first real iron meeting that I went to. It was actually the second iron meeting I went to, but the first real one uh, in San Malo. And this was in 1997. And Mark Fleming was going to talk for the first time about the identification of DMT1, then called NRAMP2, as the transmembrane iron transporter. And close to the time of the meeting, I heard that a lab just across the street from us, that at Brigham and Women's Hospital, that I didn't even know existed, had simultaneously identified a protein that they thought was a transmembrane iron transporter. And Hiromi Gunshin was there from Matthias Hedegar's lab. That was the lab across the street. And talked to her for a second at the beginning of that meeting, and said, we're going to say what the protein is. And she said that her advisor had told her not to. And I said, I think we've probably got the same protein. We talked about it enough that it seemed like we did. And that it was obviously her choice and her advisor's choice, but that I thought it would be strange. It wouldn't reflect well on them if we came out with detail about what this protein was, and they didn't at that time. And I think Hiromi made the right decision, and she did say that we had the same molecule. They came at it by a completely different approach. We did it by genetics. They did it by expression cloning. But I think that that was a lesson for all of us in a way. And later, Hiromi came over and did a second postdoc in our lab, and it was great to have her there. Two of my star postdocs over the years, Hiromi Gunshin and Joanne Levy, both died early of terrible diseases, and they're recognized by the International Bioiron Society, I think, still through travel fellowship. But both of them were really wonderful scientists. And
great to
Hiromi,
talk to. Great to
yes,
talk to. Yeah,
yes,
yeah,
yeah,
yeah. Missed in the community over the years. Way too young. Way
way
too.
too young. I think both in their 30s.
Yeah. Yeah, I agree. I agree. Okay, I'm trying to. I think that might be it, unless there's something you want to shout out about, Nancy.
I would just say I always love visiting Utah. I think I probably visited your iron folks out there more than any other iron cluster I can think of, and it was always great. Science was great, and the friends are great.
Yeah, good community. I'm sure I can say this for Jerry, because I talk to Jerry Kaplan about once a week still, that
Oh, wonderful.
he would tell you hello. And I know Betty Liebold is still here,
Oh,
and
wonderful.
still has an active lab for a few more years. And I'm sure she would want to say hello as well. Jim Kushner retired many, many years ago, lives in New
I
York
remember.
City.
Oh,
And
oh, wow.
he and Mary spend a lot of time going to the opera.
Oh, that's wonderful.
Yeah,
I know he loved it. Oh,
he did.
great.
He did. And I know Jerry talks to Mary about once a month or something like that. Jim's hearing is so bad, it's hard for him to communicate well. And so they communicate through Mary, his wife. And so they're doing really well. And I just have incredibly fond memories of the community, and also people coming here, with the history with Max Winthrop and George Cartwright. It kind
Yes.
of was this dominating area for us and stayed that way for a long time.
Absolutely, absolutely.
Yeah,
I had pictures of Wintrobe and Cartwright on some of my slides. Um, well, please give my best to all of those folks.
I sure will. And thank you again so much for this. We will, I've just so that you know, we've interviewed you, we've interviewed Harry Daly, we've
Oh,
interviewed Tom Gantz. We're going to interview Roland Lill and Jerry Kaplan. And that will be our series for this year. And then, if you have suggestions of others, Mark, of course, comes to mind, along with many, many others. If we are loving this and archiving it. I feel like it should be like in the National Library of Medicine or something.
oh,
And I'm happy to share it if that's what is decided, and they can edit it in any way they want.
well, the people who come to mind actually are European colleagues like Clara Kamaskela and Matthias Hense, and Martina Muckenthaler now,
Yep,
as well. And
those are really good suggestions.
and the Paris group, I don't, I think
With
Cajo
Pierre.
Como is longtime retired, but in Pierre, yes, absolutely.
Yeah.
It was another wonderful thing about the iron community was it was so international, and in contrast to a lot of the scientific communities I knew, it was pretty well distributed between men and women, which was
Yes.
not at the time was not very common.
Yes. Yes. And good colleagues and good friends.
Yes, yes, for sure.
Okay. Thank you
Great.
so much, Nancy. Really appreciate it. And I will let you know when we get all of this posted so that you can kind of go and look at the other interviews. And hopefully it's been a fun event.
It
And
has.
it's
And
so
it's
nice
great
to
to
see
see
you.
you.
Okay.
And nice to meet you, Cade.
Great to meet you as well.
Good luck with your thesis.
Thank you.
All right. Bye-bye.
Bye bye.
Thanks, Cade.